Welcome back, everyone. I'm Professor Piivand Pirouzi, and today we're exploring CTD Module 4 for Health Canada Clinical Trials Applications, Non-Clinical Study Reports. Module 4 contains all the preclinical studies that support your clinical development program. These studies establish pharmacological activity and characterize safety before human exposure. For CTAs, Module 4 is critically important. Health Canada will not authorize a clinical trial unless sufficient non-clinical work demonstrates that proceeding is reasonably safe. Module 4 contains all non-clinical study reports. It's harmonized across ICH regions following ICH-M4S, so the same package works for Health Canada, FDA, and other authorities. The module covers pharmacology, pharmacokinetics, and toxicology. GLP compliance is required for pivotal safety studies to ensure data quality and integrity. For CTAs, Module 4 directly supports Health Canada's assessment of whether your proposed clinical trial can proceed safely. Module 4 follows logical organization by study type. Section 4.1 is the table of contents. Section 4.2.1 covers pharmacology including primary pharmacodynamics, secondary pharmacodynamics, and safety pharmacology. Section 4.2.2 covers pharmacokinetic studies characterizing ADME in relevant species. Section 4.2.3 covers toxicology, typically the largest section, including acute, repeat dose, genetic, and reproductive toxicity studies. Section 4.2.1 contains pharmacology studies demonstrating biological activity. Primary pharmacodynamics shows your drug's intended mechanism. Secondary pharmacodynamics characterizes effects beyond the primary mechanism. Safety pharmacology assesses effects on critical organ systems, CNS, cardiovascular, and respiratory. These studies identify potential risks requiring clinical monitoring. Health Canada uses pharmacology data to understand what your drug does and what effects to anticipate in clinical trials. The Safety Pharmacology Core Battery follows ICH S7A and S7B guidelines and should be completed before first-in-human studies. CNS assessment evaluates effects on behavior, coordination, and neurological function. Unexpected sedation or excitation would be concerning. Cardiovascular assessment is critical. The HERG assay evaluates potential for QT prolongation in cardiac arrhythmias. In vivo cardiovascular studies assess blood pressure, heart rate, and ECG parameters. Respiratory assessment evaluates effects on breathing rate and tidal volume. Health Canada expects the core battery before authorizing Phase 1 CTAs. Findings directly inform clinical safety monitoring. Section 4.2.2 covers pharmacokinetic studies characterizing how the drug moves through the body. Analytical methods for measuring drug concentrations must be validated. Absorption studies characterize bioavailability and factors affecting it. Distribution studies show where the drug goes in the body, including tissue penetration. Metabolism studies identify metabolic pathways and metabolites. Excretion studies characterize elimination routes and half-life. Drug-drug interaction studies predict clinical interactions. PK data are essential for species selection in toxicology, dose selection for clinical trials, and interpretation of toxicology findings. Section 4.2.3.1 contains single-dose or acute toxicity studies conducted in 2 species. The rodent species is usually rat or mouse. The non-rodent is usually dog or non-human primate. Species selection should be justified based on relevance to humans. These studies determine the maximum tolerated dose and identify target organs of acute toxicity. Findings inform repeat dose study design. While acute studies alone don't support CTAs, they provide important preliminary toxicity information. Repeat dose toxicity studies are the most critical for CTA support. ICH-M3 guidance specifies duration requirements. Study duration should match or exceed intended clinical exposure. For single-dose phase 1 studies, 2-week repeat dose studies typically suffice. As clinical exposure extends, longer toxicology studies are needed. 1-month clinical trials need 1-month toxicology studies. These studies identify target organs, establish no observed adverse effect levels, and assess reversibility. GLP compliance is mandatory for CTA-supporting toxicology studies. Genotoxicity studies assess potential to damage DNA. ICH-S2 defines the standard battery. The AIMS test detects gene mutations using bacterial strains. In vitro mammalian assays detect chromosomal damage in cultured cells. The in vivo micronucleus test assesses chromosomal damage in living animals with intact ADME processes. The standard battery should generally be completed before phase 1 CTAs. Positive findings raise serious concerns requiring careful evaluation before proceeding. Carcinogenicity studies are 2-year rodent bioassays required for drugs intended for chronic use. They're not needed before phase 1 or 2 but should be completed before large phase 3 trials. Reproductive toxicity studies assess effects on fertility, pregnancy, and offspring development. Embryo-fetal development studies are critical before including women of childbearing potential in trials. These identify teratogenic potential. Timing of reproductive studies depends on patient population. If only men or postmenopausal women will be enrolled, timing is more flexible. Additional toxicity studies may be needed depending on drug characteristics. Local tolerance studies are required for non-oral routes. An injectable needs injection site tolerance assessment. Antigenicity studies assess immune response potential, particularly important for biologics. Immunotoxicity studies evaluate effects on immune function for drugs that might affect immunity. Mechanistic studies help understand observed toxicities. Dependent studies assess abuse potential for CNS active drugs. Let me summarize non-clinical requirements by clinical phase for Health Canada CTAs. For phase 1, you need the safety pharmacology core battery, repeat dose toxicity studies of appropriate duration, typically 2 weeks minimum, and the genotoxicity standard battery. For phase 2 and 3, toxicology study duration extends to match clinical exposure. Reproductive toxicity becomes important when including women of childbearing potential. ICH-M3 provides comprehensive guidance on non-clinical studies to support clinical development. Health Canada follows these international guidelines. Species selection for toxicology studies requires scientific justification. Pharmacological relevance means the species should respond to your drug through the intended mechanism. The drug's target should be present and functional. Metabolic similarity to humans ensures relevant metabolites are formed. A species that metabolizes the drug very differently may not predict human toxicity. Toxicokinetic measurements verify adequate drug exposure. Species that rapidly eliminate the drug may not achieve meaningful exposure. Typically, rat is the rodent species and dog or non-human primate is the non-rodent. Your species selection should be justified in the non-clinical overview. Good laboratory practice compliance is required for pivotal safety studies supporting CTAs. GLP regulations ensure data quality and integrity through documented procedures, quality assurance oversight, and archive requirements. This gives Health Canada confidence in the data. Repeat dose toxicity and genotoxicity studies supporting clinical development must be GLP compliant. Safety pharmacology core battery studies should also be GLP. Non-GLP studies are acceptable for exploratory work like early pharmacology studies, but pivotal safety studies require GLP. Include GLP compliance statements or certificates in your study reports. Clinical dose selection is based on non-clinical safety findings. This is critical for CTA review. The no observed adverse effect level from toxicology studies is a key reference point. Convert the NOAEL to human equivalent dose considering species differences. Calculate safety margins between proposed human doses and doses causing toxicity in animals. For first-in-human studies, substantial margins are expected. Health Canada, like other agencies, expects conservative dose selection for first-in-human studies. Your dose rationale should clearly explain how proposed doses relate to non-clinical findings. Inadequate dose rationale is a common deficiency leading to clarification requests. Let me discuss common deficiencies in Module 4 submissions. Inadequate study duration for clinical support occurs when toxicology studies don't match proposed clinical exposure. ICHM 3 specifies minimum durations. Missing or incomplete safety pharmacology, particularly the core battery, raises immediate safety concerns. Species selection must be scientifically justified. Unexplained species choices prompt questions. Insufficient toxicokinetic data prevents safety margin calculation. Without TK data, the value of toxicology findings is diminished. GLP noncompliance for pivotal studies is a serious deficiency. Health Canada expects GLP compliance for safety studies supporting CTAs. Let's conclude with key takeaways about Module 4 for Health Canada CTAs. Module 4 contains all non-clinical study reports organized into pharmacology, pharmacokinetics, and toxicology sections. Your non-clinical package must support your specific proposed clinical trial. Phase-appropriate studies of adequate duration are required. GLP compliance is mandatory for pivotal safety studies. Non-GLP studies may supplement but not replace GLP studies. Non-clinical findings directly inform clinical trial design and safety monitoring. Target organ toxicity should be monitored in clinical subjects. In our next and final lecture, we'll cover Module 5, which contains clinical study reports and the proposed clinical protocol. Thank you, and I'll see you next time.